Hello students, welcome to E-Pathashala. I am Dr. Vasanthi, Professor and Head, Department of Biotechnology, Kamaraj College of Engineering and Technology, Virudhunagar, Tamil Nadu. In today's module, we are going to see about lipids and most importantly, structure of lipids. Lipids are a very important class of biomolecules and they are defined by their low solubility in water and high solubility in non-polar solvents like chloroform, benzene, etc. Their composition is hydrocarbon in nature and they are highly reduced forms of carbon. However, on oxidation, they yield large amounts of energy. Lipids, as an important class of biomolecules, are indispensable to metabolism and cell structure. Based on their function, lipids can be classified into storage form and structural form, and based on their structure, they can be classified into simple lipids, compound lipids, precursor and derived lipids. Under simple lipids, we have fats and waxes, and under compound lipids, we have got phospholipids, glycolipids, glycosphingolipids, and lipoprotein, sulfolipids, and aminolipids. Now let us take the first one called the fats, which are called simple lipids. Fats are also known as triglycerides because they are formed from the glycerol backbone. Let us review our basic understanding of glycerol structure. Glycerol has CH2OH, CHOH, CH2OH, 3 hydroxyl groups, and these 3 hydroxyl groups are replaced with a fatty acid to make a triglyceride. They are the chief storage form of lipids in the body. They can be termed as esters of fatty acids with glycerol. Fatty acids are also called as carboxylic acids and they typically contain between 12 to 20 carbon atoms. They usually have an even number of carbon atoms. Try to represent the ball and stick model and the chemical structure of a fatty acid. It has a hydrophilic head and a hydrophobic tail. It's both a polar molecule and an apolar molecule, a polar head and a non-polar tail. Now, the fatty acids can be further classified into saturated fatty acids and monounsaturated fatty acid depending upon the presence of double bonds in them. Saturated fatty acids, example stearic acid, which is an 18 carbon fatty acid, does not have any double bond, it has only single bonds. Whereas unsaturated fatty acids, for example oleic acid, has double bond in its structure. Unsaturated fatty acids are again classified into monounsaturated fatty acids if they have one double bond, and they are called as polyunsaturated fatty acids if they have 2 or more double bonds. And examples of polyunsaturated fatty acids are the linoleic acid and linolenic acid. As I already explained you, triglycerides are lipids that have a glycerol backbone. 3 OH groups of the glycerol are attached to 3 fatty acid moieties through ester linkages. So the bond between glycerol and the fatty acid is through an ester linkage. Triglycerides are the main constituents of vegetable oils, which are often more unsaturated than animal fats, which are typically more saturated. Let me make it clear. Vegetable oils are mostly unsaturated in nature, whereas animal fats are typically saturated in nature. Triglyceride molecules can also be classified, as I told you, as saturated and unsaturated. The degree of saturation and unsaturation gives rise to another phenomenon called solid versus liquid triglycerides. Triglycerides with a higher degree of saturation that is less TC double bond, are more flexible and they can pack closer to each other than the less saturated triglycerides. Animal fats have a high degree of saturation and they are hence solid at room temperature, whereas vegetable and fish oils, they have a lower degree of saturation and hence they are present as liquid at room temperature. Higher the degree of saturation, more solid form, lower the degree of saturation, liquid at the room temperature. The next important class of simple lipids are called the waxes. Waxes are again hydrophobic molecules. Here there is a very long fatty acid and an alcohol. Biological waxes are esters of long chain. The fatty acid can either be saturated or unsaturated, but what is more important here is they are usually long, 14 to 36 carbon, and after that they have a long chain of alcohol which can be 16 carbon or of a 30 carbon. The fatty acids which are present in waxes are usually saturated fatty acid. Hence they have a very high melting point, 60 to 100 degrees C, and they are solid at room temperatures. They play a very important biological role as protective coatings of leaves, fruits, fur and feathers and exoskeletons of animals. So both for plant kingdom and animal kingdom, waxes are very important. And the biological waxes find a variety of application, and examples of biological waxes are beeswax, wax from the plant origin. The next important class of compound lipids are phospholipids. Very important class of phospholipid for maintaining the architecture and the structure of the plasma membrane. Here again, a phosphate group is attached to the polar head region. In phospholipids, so phosphate ion is one of the important components. Based on the structure, phospholipids can be classified into glycerophospholipid if they have got glycerol as a backbone, or they can be called as a sphingophospholipid if they got sphingosine as a backbone. Let me draw your kind attention to the diagram here. Here we have clearly shown that if it has got a glycerol backbone with two fatty acid and the third fatty acid, third position of the glycerol moiety, the OH group is substituted by a phosphate group followed by an alcohol. This becomes a glycerophospholipid. Whereas in the second picture, we have shown that the backbone is a sphingosine and not of glycerol. Still, there is a fatty acid, and in one of the group attached is a phosphate and a choline. Now let us look into glycerophospholipids. Phosphatidic acid, or commonly called as phosphatidide, is the simplest glycerophospholipid. There are two OH groups which are esterified to fatty acid, and the third one is esterified to a phosphate. This is a very key intermediate in the synthesis of other glycerophosphates. The phosphate is again esterified with another X group. Let me make it very clear. When the phosphate, one of the hydroxyl group of the phosphate can be replaced by an X group. Depending upon the presence of the other X group, a different kind of phospholipids by name phosphatidyl serine. If serine is attached to the OH group of phosphate, ethanolamine, it is called phosphatidyl ethanolamine. If it is choline, it is called phosphatidyl choline. If it is glycerol, it is phosphatidyl glycerol, and if it is inositol, it is phosphatidyl inositol. So you have got a wide variety of glycerophospholipids that can be synthesized. The next important group of lipid is called the sphingolipids. Sphingolipids also have a polar head group and two non-polar tails, contain sphingosine, which is a long chain amino alcohol. The carbon C1, C2 and C3 of the sphingosine molecules are structurally analogous to the three carbons of glycerol. Sphingosine by default contains a long hydrocarbon chain. When the amino group of the sphingosine forms an amide bond with the fatty acid, it forms a ceramide, which is structurally similar to a diacyl glycerol. Ceramide is a structural parent of all sphingolipids. like glycerosphingolipids, we have got phosphosphingolipids too. Now let me draw your kind attention to the diagram here. Let us understand the difference between sphingosine and a ceramide. In a sphingosine, the 3 carbons similar to the 3 carbons of glycerol, CH2OH, CHOH and CH2OH. The first carbon remains CH2OH, but in the second group there is an amine substitution, and then there is again a larger group of analogous compounds of sphingosine background present. So sphingophospholipid are made by combining a sphingosine, a fatty acid, a phosphate group and an alcohol. The sphingosine is called the backbone, the fatty acid forms the tail region, and the alcohol part and the phosphate part forms the head group. A sphingomyelin is a ceramide with a phosphocholine or phosphoethanolamine as their polar head. Now let me draw your kind attention to the physiological significance of sphingomyelin. They are found in the myelin layer, myelin sheath of the spinal cord, a bilayer that wraps around the nerve cell axons. Why is this sphingomyelin very important? It is a very important class of lipid to maintain the potential of the axons, nerve cell axons functions. What happens as we age? As we age, the ability to synthesize complex lipids like sphingomyelins and sphingophospholipid are reduced to a greater extent, thereby weakening the myelin bilayer sheath of the nerve cell axon. So what is going to happen because of this? Due to the weakness of the myelin sheath layer, the excitability of the nerve cell axons are also affected, leading to a loss of effective nerve cell impulse conduction. So these complex lipids, although are very significant for maintaining the structure of the spinal cord, they are also important for the functioning of the central nervous system, more importantly nerve impulse conduction. Let me now take you to a very important class of lipids called glycolipids. As the name clearly suggests, we have got lipids that contain sugar residue. Glyco means sugar, lipids denotes the lipid part. The glycolipids are again divided into 2 categories: sphingolipids, galactolipids. As in sphingolipids, you have got a sphingosine backbone, a fatty acid. Along with that, earlier, where we used to have a phosphate group and an X group attached to the phosphate, in a sphingoglycolipid, we will have either a mono or a disaccharide. I would like to draw your kind attention that because it is a glycolipid, there is a presence of monosaccharide or disaccharide and no phosphate group is present. The membrane lipids, in which the hydrophilic head groups are oligosaccharides and they act as specific sites for recognition by the carbohydrate binding protein. Glycolipids are essentially membrane lipids. I once again reiterate that because of their hydrophilic head groups and also due to the presence of the oligosaccharide moiety, they can act as recognition molecules by the carbohydrate binding proteins. As I already told you, they do not have any phosphate group, and the sugar can either be a monosaccharide, oligosaccharide or a polysaccharide. If it is a monosaccharide, the structure is less complex. If it is a polysaccharide, it is a more complex lipid structure. Sphingoglycolipids, again sphingosine backbone with carbohydrate. They are again classified into cerebrocytes, globocytes and gangliosides. Whereas the glyceroglycolipids, which originate from the glycerol backbone, can again classified into galactolipids and sulfolipids. Glycolipids, sphingoglycolipids, glyceroglycolipids. Now, let us look in detail a little more about the sphingoglycolipids. As I told you, they include cerebrocytes, globocytes, and ganglioside. What is a cerebrocyte? It is a sphingolipid, that is a ceramide, with a monosaccharide such as glucose or galactose as the polar head group. The globocytes are neutral in nature. They are uncharged glycosphingolipids with 2 or more sugars. Usually, it is a D-glucose, D-galactose, or N-acetyl D-galactosamine. Cerebrocytes and globocytes are sometimes called as neutral glycolipids as they have no charge at pH 7. The third component of sphingoglycolipids are the gangliosides, which are ceramides with a complex oligosaccharide as the head group. They are the most complex sphingolipids and have oligosaccharides as their polar head groups and 1 or more residues of N-acetyl uraminic acid, a sialic acid, which is simply called as a sialic group, and the termini. The sialic acid gives the gangliosides the negative charge at pH 7, thereby distinguishing them from the cerebrocytes and the globocytes. Now let me take you to the glyceroglycolipids. In many cases, the sugar and the fatty acid residues are attached as usual to a glycerol backbone, and hence they are called as glyceroglycolipids. Now let me take you to another important complex lipid component. So far we were focusing our attention on the fatty acyl derivative based simple lipids and complex lipids. There's another arm of a lipid structure called the complex steroidal structure. They are also a class of lipids that share the same basic ring structure. 3 fused 6 carbon atom rings and 1 5 carbon atom ring. I think this is sufficient for us to understand the complexity of the structure. 6 carbon atom ring, 3 fused 6 carbon atom ring and 1 5 carbon atom ring. There are 3 important types of steroids. One is called cholesterol. The second one are the steroid hormones which are derived from the cholesterol based structures, and the third one are called the bile salts. Cholesterol, as we all know, it's another important steroid which is found in most of the human and other animals. In cholesterol, the non-polar ring and the hydrocarbon chain are hydrophobic, and the OH groups, which are plenty in number, which makes up a much smaller part of the molecule, gives it its hydrophilic nature. Cholesterol is a very important sterol in animal tissues. It is amphipathic because it has got both hydrophilic nature, it has got a polar head group and a non-polar hydrocarbon body, which is the steroid nucleus, and the hydrocarbon side chain at C17, and which is a long carbon fatty acid chain which leads to its extended structure. Cholesterol is a very important component of the membrane along with phospholipid. Cholesterol is also the precursor of the other steroid hormones and the bile salts, which are very, very essential for other digestion and other kind of mechanisms. Steroid hormones are very important class of hormones which are involved in the secondary sexual characteristic formation and the oogenesis phase and the spermatogenesis components of human reproduction system. The bile salts and the bile acids play a very important role in the digestion process of the body. These pigments are very important for flushing away of the unused lipid entities after absorption by the intestine through the feces. Today we have tried to understand the structure of lipids. To summarize, we have got 2 important classes of lipids based on their structure: simple lipids and compound lipids. The simple lipids include the fatty acids and the waxes. The complex lipids are the phospholipids, sphingolipids, and we have got more complex lipids, to be more specific, steroids, which are on the other end of the structural complexity of the lipid structure. Lipid structures are very essential to understand the property and the function of the lipid. the structure of the lipid is invariably going to reflect on the property. There are n number of properties. As I've already told you, this is a very significant biomolecule characterized by its non-solubility in water and solubility in hydrophobic environment. It is indeed the structure of the lipid which is responsible for its properties and also for their diversified function in storage and maintenance of cellular metabolism.